Well, thank you everybody. Thank you everybody for joining. This webinar is the first of a new series of webinars from ISAC's CYTO Innovation Committee, and the theme is Next Big Things in Cytometry. And what we mean by next big things is areas of rapid innovation in cytometry technology, which have a potential to become a widespread technique in biology. We're looking at things that are beyond the current horizon, beyond the horizon of things that most cytometrists have access to, because the large cytometry companies either don't have or haven't invested in these technologies yet. But where there is a critical mass emerging of researchers who are rapidly developing these technologies and showing their potential. So the first topic of the next big things, the first next big thing, if you will, is mechanocytometry. And for that, we have an interviewee, which is Professor Oliver Otto, who is joining us to discuss it. Oliver is the Professor of Cellular Biophysics at the University of Greifswald, and he's also co-founder of Zellmechanik Dresden, which is commercializing real-time deformability cytometry, which is, well, we'll hear about it today, high-throughput method for the mechanical characterization of cells in suspension. Oliver also serves as the current president of the German Society for Cytometry. And his background is he gained an interest in mechanocytometry working with the late Jochen Guck, Professor Jochen Guck in Dresden. And, well, Oliver will say a bit more about that. Oliver has a PhD in biophysics from Cambridge University. Your interviewers here, well, firstly, I'm Samson.
I'm Catherine.
And we hope you'll enjoy our this interview with Oliver. So, first question, Oliver, is really how did you get interested in this topic of mechanocytometry? What's the story there?
Yes. So first of all, thank you very much for having me. It's really a pleasure. I told you before, Samson, I'm quite nervous, but I will try my best to share this vision about the next big thing from my point of view. As Samson already mentioned, I did my PhD in Cambridge and the Cavendish laboratory. And here I started to look at the mechanical properties of single molecules, more precisely DNA, together with my PhD supervisor, Ulrich Kaiser. And here we applied optical tweezers, to manipulate single molecules. And back in Cambridge, I already came in contact with Jochen Guck, who sadly passed away last year. And he developed also an optical tweezers-based technology to manipulate the mechanical, to look at the mechanical properties of individual cells. And after finishing my PhD, I started to talk to Jochen, and back then he was offered a professorship at the Technical University in Dresden in Germany. And he simply asked me to join. And he told me during a car ride, actually, look, Oliver, mechanical properties of cells are great. They are fascinating. They're interesting. They will be highly important for biophysics and the whole life science community. But there's one fundamental drawback, And he said the issue was that there was no technology to measure large sample sizes, to measure mechanical properties in high throughput. And he had this vision, And he basically shared this vision with me during a car ride. And I was so fascinated by that I decided to join him in Dresden in, I think, 2012.
Great,
So
yeah. And
I was going to ask, taking a step back. So you said that he convinced you that mechanics is super important, but why are they so important? What are the big questions that it can answer?
I think one of the big advantages or the big selling points is that if you consider mechanical properties as a kind of biomarker, that they are orthogonal to what you could call established biomarkers in cell biology. Yeah? If you think about fluorescent markers, or if you think about transcriptomics and genomics, and they're orthogonal in a way that they're intrinsically label free. Yeah. So for most, or for many, let's put it like this, that for many assays in cellular biology, you need to have an educated guess, yeah, what molecule, yeah, what protein has been changed, yeah. And for mechanics, you don't need that, yeah. You can have a purely unbiased view on life, and this was for me totally fascinating.
so I think it's probably a good time to ask you what is mechanocytometry, and can you give a flavor of the breadth of things that people are aiming for that can really fall into that bucket?
Yes, so as I said, so mechanocytometry, as the name suggests, looks at the mechanical properties of cells, and the best way to maybe to introduce it is by thinking about the last time you went to a supermarket, yeah, and if you're let's say you were interested, yeah, in buying some tomatoes, yeah, and you wanted to know, yeah, whether or not these tomatoes are ripe. Yeah, either you want to use them for tomato salad or to make pasta. Yeah, usually simply by looking at them, yeah, will not give you an sufficient answer. You need to touch them, you need to grab them, so you need to perform a mechanical test in the sense of a quality control. Yeah, and the point, yeah, is that mechanical testing is or has always been very something very intuitive for us human beings. And the idea is now of this whole field of yeah, cell mechanics or cell mechanical phenotyping to translate that thinking on a cellular scale. And there have been approaches since years and decades, for example, using atomic force microscopy and a micropipette aspiration. And there these technologies also contributed a lot, I would not I would say, so this technology contributed a lot to our fundamental understanding of biophysics, of cell biology, pharmacology, and so on and so forth. For example, by understanding the link between the cytoskeleton and mechanics. And I'm mentioning this because if you touch a tomato, right, you have some feeling what you will have for dinner, maybe. But if you measure the mechanical properties of cells, and if you can say whether or not a cell is soft or stiff, you would be ultimately interested into the, let's say, what is the underlying molecular um, reason, yeah, for a certain stiffness, let's say. Yeah, and the initial experiments, sorry, sorry for talking a lot. So the initial experiments in cell mechanics firstly established that link between the cytoskeleton and cell mechanical properties. And if you know reasoning that almost any physiological process or any pathological process, yeah, involves the cytoskeleton, then you can also understand that changes in mechanical properties of cells will reflect a true molecular phenotype state of a cell as well.
Yeah, you mentioned a couple of things there. I mean, firstly was the jump from, say, measuring properties of individual cells with AFM, optical tweezers, and so on, and then measuring populations of cells. So I wondered, why is that important? And secondly, well, maybe we'll stay with that one first, but then I've got another one coming up.
So you mean why it's important to measure populations of cells or large
Yes.
number of cells?
Yes, yeah. Why should there be something rich there in populations or large numbers?
So I think one fundamental advantage to have the ability to measure large sample sizes is always that you can look for subpopulations. Yeah. So if you have heterogeneous samples, and I will highlight an example later on, like blood, for example, yeah, which consists of different cell types, yeah. Having the ability just to measure a few cells might not give you a clear picture about the status of a patient, for example. Yeah, you need to measure
naturally,
really,
yeah.
not naturally, yeah, right? You need to.
Well, yeah, of course, yes. It totally makes sense. But I guess, is that what you found, for example? Is there phenotype information or say subpopulations? There are subpopulations there that are not readily identified by, say, surface antigens.
That's a good question. It's already a look into the future, right? So I think the answer would be maybe, yeah. So you could, I think there is some, I would say, there is some evidence, yeah, that, so for example, we know, yeah, that activated immune cells, yeah, change their mechanical properties, yeah. So independent. So I would not like to comment whether they are getting more stiff or more soft, yeah, but immune cells, for example, in response to a flu, yeah, to an activation of the immune system, change their mechanical properties, yeah. And having the ability, yeah, to measure large sample sizes, yeah, you could now also, and maybe also to sort these cells, yeah, you could ask a question if every, let's say, mechanically altered immune cells, yeah, after an activation, yeah, also has the corresponding surface marker, yeah, which reflects an activation of the cell, and vice versa, yeah. And this might give you an additional, let's say, perspective on a specific set of subpopulations of cells which we might not know so far. Yeah. So I'm always saying it's not my goal to do any, let's say, to say mechanics is better than cellular or molecular biology. Yeah. But I think what we would like to propagate is the idea that cell mechanics is a true orthogonal biomarker, yeah, which can provide us additional information, ideally, yeah, in combination with molecular biology.
Well, the other thing there is, is it a single biomarker there, or is it a set, or is it a rich little set, sort of expanding perhaps even of biomarkers? And how is it? Because of course the biologists generally are all thinking about molecules and signaling pathways. Is it possible to connect the two? Um, I mean. Course, ultimately it ought to be possible, but what's the how rich is that space of the connections between the structure microscopically and the measurements that it's possible to make?
So I think it's very rich, and in fact, we are learning almost every day something new. Yeah, I mean, I guess this is true for almost everybody in the audience. This is why we are scientists and why we love being scientists. So from a very basic point of view, yeah, I could start saying, okay, a cell is soft or stiff. Yeah. However, even this is not easy because it always depends on your reference range, and having an appropriate reference range is also a very important point, which we could discuss later. And from a physicist point of view, yeah, so you would say, okay, stiffness, it's not a very clear measure. You could talk about elasticity and viscosity. but in fact, if you now look more and more on what we started to call the mechanical properties of cells, as I said, we learned that you in fact have a variety, yeah, of multiple properties we can look at. Yes, we can look at the size of the cells. Yeah, we could, for example, if you think about cells in suspension, we could look at the curvature. We can look at the brightness of the cells, at the brightness distribution, which corresponds a little bit to the sideward scattered signal in fluorescence-based flow cytometry. And of course, taking or considering the fact that what we do, at least in our approach towards mechanocytometry, because what we do is we do high throughput video microscopy. So we have the big advantage that we have access to an image of every individual cell we analyzed, yeah, then artificial intelligence is more or less obvious, yeah, to having more insights, to maybe not to understand, but to classify cells even further.
Right. Yeah. Yeah. I mean, the connection to that is very clear, and obviously, it's a good time to be taking advantage of that kind of data, right?
Exactly, and to your second question, yeah. So how to link, yeah, mechanical properties and let's say the molecular state of a cell. I mean, what's very, let's say, what you always do, or what we started with, yeah, when we introduced a new technology, is to say, okay, we have to interfere with the cytoskeleton. Yeah, that's the zero level test. What you have to do, you have to manipulate with filamentous actin or microtubuli, because if this, yeah, manipulation does not induce any measurable change in mechanical properties of your cellular model system, yeah, then you might have a problem with your technology. But thinking beyond, yeah, you could also ask questions, okay, is it only the cytoskeleton, yeah, that contributes to the mechanical properties of cells? What about all the organelles? Yeah, I mean, the nucleus is a rather big organelle in the cell. What about the cell membrane? Yeah, what about the interplay about the cell membrane and the cytoskeleton? And this is all so far being how to say, true for cells in suspension, yeah. But what about if you think about cells in more like a tissue-like context? Yeah.
Yeah. Right.
About
Yes.
the mechanical changes that you can induce in cells and that you can also observe, as you mentioned, with the immune activation. Do you know, are these mechanical properties of cells, are they generally fulfilling a purpose for that cell's function? Or do you think it can also just be a kind of byproduct of the cells changing their molecular state?
So I would say either or, yeah. And that's it's a really good question. So a few years back, for example, we looked at the infection of red blood cells by a malaria parasite. Yeah, and this was quite an exciting time. We went to Glasgow, and we let's say were allowed to measure blood samples that have been infected by the malaria parasite. And interestingly, what we observed is that infected red blood cells are stiffer after infection. And what our collaborators then also could show is that the parasite basically after infiltration locally alters the mechanical properties of the cell membrane. Yeah, and this inhibits, or this basically, if once one parasite entered a red blood cell, the mechanical properties of the cell membrane are altered in a way that not a second one is not able to infiltrate. Yeah, so there's one example where there's also where mechanics, or this is one example where mechanics is related to function. Or if you think about immune cells, yeah, and the activation or deactivation, priming, depriming of immune cells. There are some studies with from, I think, the University of Cambridge, where also back then in collaboration with Jochen Guck, where people were interested in what's happening when red blood cells pass our lung capillaries. And apparently, yeah, there's evidence that if our immune cells squeeze through lung capillaries, yeah, that this, let's say, massage, yeah, is also responsible for, or it's one way to deprime immune cells, yeah, which should stop, for example, also an overreaction of our immune system. And this is another, let's say, a link between, let's say, functioning, yeah, of individual cells, yeah. And if you think about chronic inflammation in our humans, so it's not only affecting individual cells, but us as human beings in general, yeah, and the mechanical properties of cells.
Um, well, I think this is, I mean, these are really good examples, and of course, the applications, what's possible with this is, this is key to, this is really important to making this a widespread technique, I think. I mean, the ones you highlighted are interesting because I mean, the mechanics is not, it's not just something you measure, but it's something uh, that you have evidence that it's actually part of the function, right? That's really, I mean,
Yes.
that's really interesting. It's mechanical measurement and mechanical functions. I guess there may be a lot of cases where mechanics may be correlated to something, but not directly you know, not related in any known way to function. I mean, could you give us a breadth of what might be possible out there in applications? Um,
So I think if you so you mean in future also?
oh yeah, yes. So I suppose
Uh,
things that are proven, things that are suspected, and things that you hope for.
So I think one thing could be that mechanics could be a good way to, as I actually motivated, or as I motivated this discussion at the beginning, as a quality control tool. Yeah, for example, if you think about tissue engineering, and here we collaborate with the German Center for Cardiovascular Research, and they create, for example, artificial heart tissue, yeah, derived from induced pluripotent stem cells, yeah. And if you think about a transplantation, yeah, of this engineered heart tissue, yeah, um, you think about, or you need to consider that this heart tissue must be able to work like your own muscle cells. You need to contract and relax, yeah. And here it's very apparent, yeah, that mechanics will play a highly important role, yeah. And maybe in the future, yeah, it could be well possible, yeah, that cell mechanics will tell you whether or not, yeah, a tissue or yeah, a tissue is suitable for transplantation. Yeah, so maybe in the future, yeah, this would be an application, or you can think about to like a mechanical based blood test to monitor a therapy, yeah, or a treatment, yeah. If you have, for example, or if you suffer from a chronic infection, yeah, chronic inflammation, yeah, like rheumatism, or if you suffer from sepsis, yeah, you could think about cell mechanics as some kind of yeah, blood test, as like a minimal invasive blood test, yeah, whether or not your treatment is effective. Yeah, and maybe I forgot to tell this at the beginning. If you consider that, so I told you that there's an interplay between the cytoskeleton and the mechanical properties of the cells. On the other hand, dynamics of the cytoskeleton takes place on a millisecond time scale. Yeah, and now Samson is gone. Can you still hear Catherine?
Yeah, I can.
Ah, okay, no, Samsung was gone. Um, so And if you now consider that you have for example, polymerization and depolymerization of actin on a millisecond time scale, you could also envision that cells react on these time scales, yeah, to different environment, yeah, and this might help you, as I said, also to monitor whether or not a drug treatment or therapy is effective, yeah, in a very, let's say, minimal invasive way.
I'm not for Samson, we can't hear you, but
Oh, sorry.
Oliver, I was thinking this would be a good time. I think we've really learned a lot about the background of mechanocytometry and why it's so important and exciting. But we'd love to hear more about your specific technology in this area. So it'd be great
Yes,
to see the slides and your
yes, yes.
talk.
So I will start sharing some slides. So I already mentioned a few of the points I wanted to raise, but let me start. So can you see my slides?
Yes.
Yeah. Yeah,
Yes.
let me start again by taking the opportunity to remember Jochen Guck, who sadly passed away in October last year, who was not only a mentor and a friend of mine and my postdoc advisor, but I would like to say was one of the big masterminds, yeah, of the entire field, and he was one of the pioneers in this field. Not only because he had a great ideas about technologies, but also he was being a physicist, he was able in a very unique way to relate biological questions, yeah, which were there, yeah, to physical or to methods which we could develop in physics, yeah, because usually we physicists are really good, yeah, in developing methods, yeah, but then we need the biological question, yeah, and he had the deep insight and the deep knowledge really to do it also the other way, yeah. He knew, yeah, that there was a need for mechanical phenotyping, and then he came up with the technology. So having said that, yeah, I hope, I hope I could already convince you, yeah, that mechanical testing is important on a macro scale. Also on a micro scale, I also told you that the big advantage of this approach is that almost any physiological or pathological process you can think about involves the cytoskeleton. And this is what we probe by looking at the mechanical properties of cells. And I already told you that we are, of course, not the first one. Yeah, there have been technologies before there, like atomic force microscopy, where you basically use a cantilever to indent cells, and if the cell is very stiff, this cantilever, it's like a spring, will bend a lot. If the cell is very soft, it won't bend at all. You can think about micropipette aspiration, where you suck a fraction of a cell into the orifice of a pipette. If the cell is very soft, you suck in a lot. If the cell is very stiff, you suck in a little. So this has all been established, in fact, decades ago, and contributed also a lot to our fundamental understanding of life. But what has been missing, yeah, was a technology, as I said, to measure mechanics and high throughput. And when I joined the group of Jochen, our idea was to come up with a technology to measure mechanical properties of cells with the throughput of a flow cytometer. And our answer, yeah, to this need, yeah, what we thought was out there, is real-time deformability cytometry. And this technology is beautiful because it's simple, yeah. And it's like a flow cytometer. So you have a capillary, yeah, or in our case, a microfluidic channel, which is basically attached to a syringe pump or a pressure-driven pump, and you pump a suspension of cells through this central channel. And then the idea is that it's like in a river, yeah. A river flows fastest in the center and slowest at the shore. And what we do is basically by shrinking this river to micrometer size, we utilize this velocity difference, yeah, to deform cells. And this is also shown here as a sketch at the top and as an time series at the bottom, where you have an initially round cells basically deforming in this bullet-like shape. And this real-time deformability cytometry is now a technology which is able to perform image analysis at 4,000 frames per second. So I told you earlier that at the end, what we do is high-throughput microscopy. And by using this approach, we can analyze more than 1,000 cells per second in real time. Yeah? And what we do is we use image analysis to detect the presence of the cell in this capillary, on this channel, and by relating cell area to cell perimeter, we can come up with a very simple deformation measure and also extract multiple parameters. Yeah? And then what we get at the end, yeah, is a plot like that, and the whole, let's say, cytometry community, yeah, should be familiar with that. Yeah, so we have a scatter plot. Instead of having a fluorescent marker or sideward or forward scattered signals, we have, for example, deformation versus cell size, where each dot corresponds to a single mechanical measurement. And I guess for the community, yeah, measuring or the ability to measure 1,000 cells per second might not seem a lot, yeah. But compared to anything else which has been there before, we are a factor of 10,000 faster, yeah. And Jochen always said we can now, I forgot, but he always said we can now measure in a day, which took 10 years before. Yeah? And this was really what I would call, or what we called the disruptive technology for the field, yeah, for the field, yeah. And based on that, we could approach now a number, let's say, of questions in microfluidics, life science research, and cell and tissue mechanics. I know these slides are made to show off, but I would like to make a very important point again, yeah, is that I also have a financial interest, yeah, and which I would like to disclose again. So I am also co-founder of. Sorry, Martin, I know you're in the audience, but I am a co-founder of probably the greatest company in the world for, let's say, developing and distributing technologies to measure the mechanical properties of suspended cells. So please, whatever I've said so far, whatever I'm going to say, take it with a grain of salt, specifically if I'm too enthusiastic about it. Okay, so having said that, so I thought to share maybe one example what you can do with this technology, because a few years ago I was approached by a friend, yeah, and or a colleague, and he told me, well, Oliver, I'm a big science fiction fan, and I would like to go to interstellar space travel, and we need human hibernation, and please help me. And then I looked at him and told him, well, Gerald, good point. I'm also a big science fiction fan, but why me? Yeah. And then he explained a bit further, and he told me that he's doing research on bats, and that he's very much interested into the ability of bats to go into hibernation, or maybe to make a bit more clear, to control their body temperature, let's say between almost let's say 4°C and 36 degree zero. And one question which really fascinated him, or what is still fascinating him, is why are some species able to control their core body temperature, and we humans, for example, are unable to do that? Yeah. And apart from going to space, yeah, um, having this ability as human beings would be absolutely amazing. Yeah, think about surgeries, yeah, where controlling our body temperature, or the ability to control our body temperature, would be absolutely helpful. So we discussed, and to cut a long story short, we came up with an idea that maybe the blood of or the blood cells of bats have some specific property, like a lubricant, yeah, which make them flow better at low temperatures. Or to say it maybe a bit more scientifically, we were interested in the question if unique viscoelastic properties of red blood cells allow for an adaptation of bats to different temperatures. Yeah, and this is a really cool topic. And what we did is we performed, I would say, a small study. Yeah? So we measured mechanical properties of non-hibernating species, humans, of a non-hibernating bat species. So we looked at so-called Egyptian fruit bats and also at a hibernating common noctule bat. And it's really interesting if these bats were brought into our lab, and we very well cared about animal welfare. To also, this is very important point, because all we needed is a small blood donation of 20 microliters of blood. So the bats and the humans, they were all happy about that. So it's not enough. And after we took this very tiny droplet of blood, the bats and the humans were released into their work business or in their daily life. Yeah, and just to make to make the final point, what we found. So yeah, I forgot. So this is when we put blood in our device, and to to make again this point about the power of an label-free cell analysis, yeah, simply by deforming cells, simply by looking at the mechanical properties of cells, we can very easily identify at least in whole blood at least 4 different populations. So the most abundant cells are red blood cells. We have platelets, and also the two leukocyte populations. We can look even further into the different lineages. I don't want to talk about that, but for this project, we have been interested in red blood cells. And one result we gained is, and this was for me really fascinating, is first of all that red blood cells of bats have, as a cell, I'm not talking about blood plasma, as a cell have a higher viscosity, which seems to be a bit counterintuitive, because higher viscosity would mean they're more honey-like, so they don't flow as well. But if you think about it, that if the viscosity is higher, maybe yeah, they have more time in the lung than at low temperature for example, for oxygen exchange. Yeah. On the other hand, we can also show that the red blood cells of bats at low temperature have reduced membrane tension, which acts as a which might act as a lubricant. Yeah, and this means that maybe bats have the best of two worlds. Yeah, they have, let's say, higher viscosity, so they spend more time in the muscles in the lung for oxygen exchange, but they also flow better. For me, it's really, really amazing. And the second big result we got is that these changes, yeah, cannot be explained by environmental factors, because it's often believed that's the diet, yeah, that the nutrition is, yeah, but we can show it's not, yeah, so it must be something really intrinsic in the mechanical properties of red blood cells of those bats. Yeah, so, and this is just one example I would like to highlight to show the power of cell mechanics. Yeah, because we didn't have any educated guess about the molecular target. Yeah, but simply by squeezing, massaging red blood cells, yeah, we could learn a lot. Yeah, and now the interesting question would come: okay, what are now the molecules responsible for these specific adaptations? Yeah.
So, well, thanks very much for that presentation, Oliver. Um, and so now we've covered what is mechanocytometry. We've covered some of the applications. We've covered your particular approach of real-time deformability cytometry. And I suppose, I mean, there are many questions, and the last sort of topic for us is what would it take for this to become a major technique in biology and medicine? So, so we'll discuss that now. But I just want to invite the audience. Please write your questions for Oliver, and we'll have some time at the end, and we'll read them out. So please go ahead and write them in the Q&A box. I suppose we could just start with we could start with this. I mean, we could discuss the bads, but maybe we better start with the big question. What
What will it
what would
take?
it take to become a major technique in
So,
biology and medicine?
so I think one thing which what the field really needs is it need to be able to make, let's say, results comparable between different laboratories, yeah, and also between different techniques, yeah. So cell mechanics is so mechanical properties of cells is a very sensitive biomarker, yeah. But this also means that it makes a difference if you, for example, indent a cell for a second, a minute, or just a fraction of a second, yeah. So what you get will be different. Yeah, so you need to find standards. You need to make sure that you can compare results between different laboratories. And talking about that, you will also need, I think, if you think about an application in a medical setting, yeah, in a hospital, you would need reference ranges, yeah. If you think about blood, yeah, or blood cells, yeah, what is a typical stiffness, yeah, of a neutrophil, yeah, and what is it age dependent? Yeah, is it sex dependent? Yeah, is it depending on your diet? Yeah, I think this is something we still need to establish. Yeah, something which is we still need to explore. And if we have that, I think there will be great opportunities really for an application as a biomarker, for example.
One thing I was wondering about that you alluded to, actually, I mean, connected to these sort of references, I mean, I suppose is you've got a technique that measures something, a phenotype of cells, in many cases related to the cell's function. And biologists, it's still very far from the sort of fundamental the theories of biology. I mean, the fundamental dogma and so on. So is there a problem that there's this, I mean, not only the values, but say the theory connecting the two? I mean, the lack of something to explain what does it mean, molecularly, if stiffness goes up and down, say models of the cell that may connect it to molecular behavior. Is that needed for the biologists to really latch onto to this in a sort of more general way?
Yes, so I think that this is needed. For example, there's evidence that, for example, when you look about the differentiation of cells into for example, of induced pluripotent stem cells into a specific lineage, yeah, the mechanical environment, yeah, plays a huge role. Yeah, so there's a paper from, I think, Dennis Discher from more than 20 years ago who showed that, let's say, if you put a stem cell, yeah, on a substrate which has more the stiffness, which has a stiffness which is more bone-like, yeah, then the probability, yeah, and differentiating into a bone cell, yeah, is much higher than into any other lineages, yeah, and we still need an understanding, yeah, what's about this interplay, yeah, between, let's say, mechanical properties, then mechanical sensing as something which I haven't talked about at all today, and about the key molecular players. Yeah, and I think it will be really helpful for, let's say, both communities, yeah, cell biologists and classical physicists like me. Yeah.
Yeah, and one thing that I found kind of enlightening about it related to this field is that, say, many of the surface antigens that, say, taken for granted as have some some meaning about the phenotype of a cell and its behavior, say in immunology. There are also molecules that are related to the cytoskeleton, like integrins or signaling molecules, or that there's that link. But that's kind of not. I mean, people are not aware of that, and yet it suggests that there may be something there if one could only look at the mechanical properties related to those molecules.
Yes, so people look at that. Yeah, so this is really what people what people are heavily researching. It's not from our research group. It's not our main, let's say, focus area. But if you look at the field, there's plenty of work and really important and intriguing work. For example, what is the role of integrins? Yeah, what's going to happen if you manipulate with, as I said, actin, also filamentous actin, microtubuli. Think about the whole family of intermediate filaments. Yeah, so people are heavily researching at that, and also the implications for cell functioning. Yeah, on those manipulations.
Yes, yeah. Is there another angle on what would it take? Are there technical advances that could, I mean, say in your field that could open up new or deeper data or more accurate data or higher resolution data, say, and could that, I mean, so for example, if you're looking at length scales, I mean, and you're currently looking at the whole cell, if you could look at the on the micron level or submicron level in the cell and get a lot more information. I mean, what might that give? I mean, or is it? I mean,
No, it's a great question. So for example, I mean, I mentioned earlier, or I asked the question, what about the role of cellular organelles? Yeah, for mechanics, but maybe in a broader context for cell functioning. Yeah, it is well known, for example, that the fission and fusion of mitochondria, yeah, is basically essential for their proper functioning. And there are specific pathologies where these processes are inhibited. Yeah, I know you could also ask the question, okay, is it what? So if you have two mitochondria fusing, or the ability or the disability to fuse, yeah, is this maybe also related to the mechanical properties of cells? Yeah, and if of these mitochondria, excuse me. And then again, in the second turn is if you could measure that, if you could show that, ideally intracellular, yeah, which is would also be a next big thing, yeah, to look at. If you could show that intracellular, then you could again ask the question, okay, knowing that, yeah, and having maybe an educated guess what is responsible to change, for example, the membrane properties, yeah, of mitochondria to inhibit, for example, fusion processes, yeah. Maybe then you could find some molecular targets also as, or some molecules as drug targets, yeah. But I think this, we are still very often on a fundamental, on the side, or on where we look at fundamental research questions. Yeah, the whole field is looking for an clinical application. We are moving towards that, but I think there's still plenty of work to do.
it's yes. So, I'm sorry, Catherine.
I want to say on the subject of clinical applications, we have a question from the audience from Tamara Pigabu-Kuster that you mentioned sepsis earlier. So would septic patients have the detectable mechanical blood cell changes that you could use for faster diagnostic processes?
Yes, that's a very good question. I just saw it in the chat. So there is we in our research group, we did not look systematically into the effects of sepsis onto the mechanical properties of cells, of immune cells, for example. However, so they change, yeah. The if you think about it, yeah, for sepsis or from patients suffering from sepsis, one very important question to answer would be if you could detect these changes very early. Yeah, because once you have this sepsis and once you have the symptoms, you are in trouble. So if you could contact, if you would have a mechanical biomarker to pick up these changes or the onset of an sepsis very early, this would be great. And there's a company, in fact, in California, which is a spin-off from UCLA. It's called CYTO, and they so I think, to the best of my knowledge, they started. So it's a microfluidic based technology looking at blood samples from sepsis patients, and they underwent a massive clinical trial, yeah, to really to look at these questions. So can you detect an early onset, yeah, of sepsis, yeah, to really to help patients? And they have now a device, yeah, which I think which is available on the market, yeah, and which is used for this purpose. However, I think it's not mechanics only. So to the best of my knowledge, they're also using an AI-based multi-parameter approach, still label free, yeah, but not only looking at the mechanical properties of cells. So in fact, this might be one clinical application which is already out into the market. Yeah. Good question.
While we're on the audience questions, I'll ask a second one from Anthony Chatterton, who wants to know if you've tested any drugs or chemical-induced effects with RTDC and what the scope of this is.
Yes, so so many. Yeah, so you so any it's a good question. Yeah, so when we basically came up with this technology RTDC, we used any possible drug that can interfere, which is known to interfere with the cytoskeleton. As for example, we looked at cytokeratin D for filamentous actin. We looked at latrancolin, B also filamentous actin. We looked at nocodazole. We looked at nocodazole, jasperkinolide, Taxol. Yes, we did. And one, maybe to have one comment on that. So one interesting finding here was that so usually drugs that interfere with the actin or actin cortex or stress fibers work very well. Microtubuli are sometimes a little bit, let's say, more challenging and ones that cells do not react so much to changes in microtubuli. And one explanation to that might be that cells in general are very good in compensating. Yeah, so they don't like to be brought out of their comfort zone, which is in fact good for us. So if you intervene with microtubuli, for example, then they just upregulate filamentous actin. Yeah. So, but the answer is yes. Yeah.
So another question from the audience, and we have time for a couple more. So do write in your questions, if you have any. From Natalia Bernans-Knoll. Firstly, fascinating talk. Thank you. I work in liquid biopsy field, and actually, mechanics of blood is a huge issue for tumor progression. Is there any model which can mimic transit of tumor cells from primary tumor mechanics to blood mechanics?
Oh, this also thank you very much for the question. It's a very good question. I would say there is, yeah, but honestly, I'm really sorry, I don't know. Yeah, so I know that there are model systems which would induce EMT. Yeah, and there are plenty of research groups looking at that, but I'm really sorry, I have not a clear or very specific answer to this question. So also in the light of so there are many research groups are, for example, also interested in, I mean, maybe this is also what the question is referring to, circulating tumor cells, right? And one of our biggest advantages, yeah, of our system is the small amount of blood we need. Yeah, I told you 20 microliters, sometimes even 10. And but on the other hand, if you, for example, look now, if we would look for circulating tumor cells, our throughput measured in total volume, so to measure milliliters of blood, we need too long. Yeah, so this is not our focus. But there are, for example, research groups in Singapore, so CT Lim, for example, who work at that. Yeah.
Another question from Luis Martinez. Have you got into the genes or proteins involved in the characteristics of the bat blood cells?
Not yet. Good question. Not yet. And here it's really also again a very good question. One thing I did not tell about this bat story was that and we also, so I just mentioned very briefly, so we observed differences also in the membrane properties between, let's say, human red blood cells and bat red blood cells. And one thing we would also be interested is the specific lipid composition. Yeah, but this is now the next thing we are aiming for. Yeah.
So I think that's all the audience
Thank you very
questions
much
we've
for
got
the
so
questions.
far. Yeah, I suppose I mean I've got a question on my mind to wrap it up. I don't know if Catherine, you got any more as well. But I suppose my question is, I mean, you're doing research on I mean, you're applying it to biology yourself, of course. But say in terms of advancing the technique and pushing the technique, is there any particular thing that you are working on next that will be purely for the technique itself?
So I mean, there are some obvious, let's say, not obvious is not a good word, yeah, but let's say some things we work on or we have established, for example, sorting based on not only mechanical properties, but also, let's say, on biophysical properties. And one thing which we are very much interested in is the question in about the contribution of, let's say, the cell membrane, yeah, on the mechanical properties of cells. This is not so easy to answer. And in fact, we are also working on a technology that would allow us, for example, to measure the mechanical properties of mitochondria, so of intercellular organelles actually inside the cell. And this will be, I think, very exciting basic and also translational research. Yeah.
Yeah. Well, I think, I mean, say this it was alluded to several times, but this intracellular aspect and organelles and deeper microscopic properties is surely going to find a lot of applicability.
And I mean, there's also, if you think about, there's now an or increasing field of research group working on artificial cells. Yeah, and also here, I think mechanics will play a great role. Yeah, you could, for example, ask the question, okay, how do transmembrane proteins, for example, change the mechanical properties of a lipid bilayer in a cellular context? Yeah, for let's say if you take biological cells, this question is not so easy to answer because you have always, I mean, you have plenty of proteins, transmembrane proteins in your
makes a lot of
bilayer.
sense.
Exactly.
Makes a lot of
But
sense
if
to
you have
look at that.
an artificial system which you can basically build from bottom up, I think you can also learn a lot. Yeah.
Yeah. So we're getting to the time limit. Well, I mean, I hope you enjoyed it as much as I did, and I hope
Thank you very much.
quite a sizable audience today. It was really good. I hope everybody enjoyed it. So we're continuing with this series. Actually, I mean, I just mentioned. Firstly, thank you to Oliver, and
Thank you, both of you.
thanks. The next one is going to be in June, 24th of June, which we're going to cover 3D imaging cytometry with Kevin O'Halloran, the co-founder of ZOMP. In the meantime, of course, is ISAC's main conference, CYTO, in Miami, 6th to 10th June. So just a plug for that. Hope to see many of you there. Yeah, thanks again. Thanks again to Oliver, and thanks for the questions today.
Yeah, also thank you for all the questions and for listening. Thanks a lot. See you.
So better, I also mentioned the recording will be posted on YouTube shortly.